Imagine your genes as the blueprint of a house. This blueprint has all the instructions needed to build and maintain the house, from the number of rooms to the type of roof. Now think of epigenetics as the interior design and furnishings of the house. While the blueprint provides the essential structure, the interior design can change and adapt based on preferences and needs, making the house unique and functional. What is epigenetics? Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Unlike genetic changes, epigenetic changes are reversible and do not alter the DNA sequence, but can change how your body reads a DNA sequence. How does it work? Your DNA is wrapped around proteins called histones, forming a structure like beads on a string. Chemical tags can attach to your DNA or histones, influencing how tightly or loosely the DNA is wrapped. If the DNA is tightly packed, the genes are less likely to be read and expressed. If it's loosely packed, the genes are more accessible and likely to be expressed. These chemical tags are influenced by various factors, including your environment, diet, stress, and lifestyle. Analogy: light switches in a house. Think of genes as light bulbs in a house. The DNA sequence determines where each light bulb is placed. Epigenetics is like the light switches that turn these bulbs on or off. Just as you can turn lights on and off depending on your needs, epigenetic changes can activate or deactivate genes in response to environmental cues. Examples to make it attractive: nutrition. What you eat can influence your epigenome. For instance, a diet rich in fruits, vegetables, and whole grains can lead to positive epigenetic changes, promoting health and longevity. On the other hand, poor nutrition can lead to negative changes, increasing the risk of diseases. Stress and mental health. Chronic stress can leave epigenetic marks that might affect mental health. This explains why prolonged stress can lead to changes in behavior and increase the risk of mental health disorders. However, positive lifestyle changes, such as regular exercise and mindfulness, can help reverse these changes. Generational impact. Epigenetic changes can be passed down from one generation to the next. For example, studies have shown that children of parents who experience famine or severe stress may have different health outcomes compared to those whose parents did not experience such conditions. Twin studies. Identical twins have the same DNA, but their epigenomes can differ based on their individual experiences and environments. This can explain why one twin might develop a disease while the other remains healthy. Real-world applications. Epigenetics has vast potential in medicine and healthcare. It can help us understand the development of diseases like cancer and Alzheimer's and lead to personalized treatments based on an individual's epigenetic profile. Moreover, it highlights the importance of a healthy lifestyle and environment in preventing diseases and promoting overall well-being. Epigenetic mechanisms are closely linked to the development of various diseases, including cancer and mental health disorders. These alterations can activate oncogenes or silence tumor suppressor genes, contributing to disease progression. For example, abnormal DNA methylation patterns are often observed in cancer cells, where they can inhibit the function of genes that normally suppress tumors. In mental health, epigenetic changes can influence brain function and development, potentially leading to conditions such as depression or schizophrenia. Recent advances in technology, particularly CRISPR-Cas9, have opened new avenues for editing the epigenome. This technology provides the potential to correct harmful epigenetic changes, offering hope for treating diseases like cancer more effectively. For instance, scientists are now able to specifically alter the epigenetic marks on genes in cancer cells, reactivating genes that can stop the cancerous growth. The field of epigenetics is burgeoning with possibilities. Future research is focused not only on understanding the basic mechanisms, but also on how these can be manipulated to prevent or treat diseases. One area of intense research is the role of epigenetics in aging and the development of age-related diseases, with the aim of finding interventions that promote healthy aging. To truly appreciate the impact of epigenetics, it's important to understand its historical roots. The term epigenetics was first coined by British embryologist Conrad Waddington in the early 1940s. He proposed that genes could be influenced by their surroundings, an idea that was revolutionary at the time. Fast forward to the 1980s, when researchers like Holliday and Arthur Riggs further defined the concept. They discovered that chemical modifications such as DNA methylation could turn genes on or off without altering the underlying DNA sequence. This was a pivotal moment, solidifying the idea that our genetic expression could be modulated by external factors. Another monumental figure in this field is Dr. Adrien Bird, who identified the role of CPG islands in gene regulation. His work has been instrumental in understanding how epigenetic mechanisms can be inherited across generations. These pioneering scientists laid the groundwork for modern epigenetics, opening the door to a new era of genetic research. Their contributions have not only expanded our knowledge, but also paved the way for innovative treatments and therapies. As we continue to explore the intricate dance between our genes and the environment, we stand on the shoulders of these giants, ever closer to unlocking the full potential of epigenetics. While diet and nutrition play a significant role in shaping our epigenome, another crucial factor is often overlooked: stress and mental health. Chronic stress has been shown to lead to epigenetic changes that can affect our overall well-being. For instance, research has revealed that sustained stress can alter DNA methylation patterns, particularly in genes associated with the body's stress response. One notable study involving Holocaust survivors and their descendants found that the trauma experienced by the survivors led to measurable epigenetic changes. These changes were passed down to their children, affecting their stress hormone levels and overall mental health. This phenomenon is known as transgenerational epigenetic inheritance. Additionally, studies on individuals with PTSD have shown significant epigenetic modifications in genes related to neuroplasticity and stress regulation. These changes can contribute to the persistence of PTSD symptoms and influence the individual's ability to recover from trauma. Depression and anxiety are also linked to epigenetic alterations. For example, decreased levels of brain-derived neurotrophic factor, BDNF, a protein crucial for brain health, have been associated with epigenetic modifications in people suffering from depression. Interestingly, positive mental health practices like mindfulness and meditation have been shown to reverse some of these adverse epigenetic changes. Regular meditation has been linked to beneficial modifications in genes related to inflammation and stress response, highlighting the potential for mental health interventions to positively influence our epigenome. Understanding the intricate relationship between stress, mental health, and epigenetics underscores the importance of holistic approaches to health and well-being. As we delve deeper into this field, it becomes clear that managing stress and nurturing mental health can have profound and lasting effects on our genetic expression. This knowledge not only empowers us to take proactive steps towards better mental health, but also opens new avenues for therapeutic interventions targeting epigenetic changes induced by stress and psychological trauma. As research continues to evolve, we can look forward to a future where mental health care is deeply integrated with our understanding of epigenetics, paving the way for more effective and personalized treatments. Pause 2. S. As we delve deeper into the realm of epigenetics, it's crucial to consider the ethical implications and societal impacts of this rapidly advancing field. The potential to modify the epigenome raises profound questions about the boundaries of science and the moral responsibilities that come with such power. For instance, the ability to alter gene expression to prevent or treat diseases is a remarkable prospect, but it also brings forth concerns about genetic privacy and the potential for misuse. One major ethical consideration is the concept of epigenetic equity. As epigenetic therapies and technologies develop, there is a risk that access to these advancements could be limited to certain socioeconomic groups, exacerbating existing health disparities. Ensuring that these breakthroughs are accessible to all members of society, regardless of their economic status, is a vital challenge that must be addressed. Pause 2S. Moreover, the potential for epigenetic modifications to be inherited by future generations raises questions about consent. Can we make decisions that will affect the epigenetic makeup of our descendants? This issue underscores the need for robust ethical guidelines and regulatory frameworks to govern the application of epigenetic interventions. Another concern is the possibility of epigenetic enhancement, using epigenetic modifications to enhance human abilities beyond what is considered normal or necessary for health. This could lead to ethical dilemmas around what constitutes fair and ethical use of such technologies, as well as the societal implications of creating enhanced individuals. It is also essential to consider how epigenetic information is used in fields like insurance and employment. There is a risk that epigenetic data could be used to discriminate against individuals based on their predisposition to certain diseases or conditions. Protecting individuals' epigenetic privacy and preventing such discrimination is a critical ethical issue that must be addressed. As we continue to unlock the secrets of the epigenome, we must balance the pursuit of scientific knowledge with the need to protect individual rights and ensure ethical practices. The potential benefits of epigenetic research are immense, but so too are the ethical challenges. By engaging in thoughtful and inclusive discussions about the ethical and societal implications of epigenetic research, we can navigate these challenges and harness the power of epigenetics for the greater good. While the scientific community continues to explore the vast potential of epigenetics, real-world case studies provide compelling evidence of its profound impact on human health. Let's delve into some remarkable instances where epigenetic changes have had significant health implications. One of the most well-documented cases involves the Dutch hunger winter of 1944 to 1945. During this period, severe famine struck the Netherlands, and pregnant women who experienced extreme malnutrition gave birth to children who exhibited significant epigenetic changes. Decades later, studies revealed that these children, now adults, were more prone to metabolic diseases like diabetes and cardiovascular conditions. The famine had left a lasting epigenetic imprint, highlighting the critical role of prenatal environment in shaping long-term health outcomes. Another striking example comes from the agouti mouse model, which has provided valuable insights into how diet can influence epigenetics and health. In these mice, the agouti gene affects coat color and obesity. Researchers discovered that feeding pregnant agouti mice a diet rich in methyl donors such as folic acid, vitamin B12, and choline could alter the expression of the agouti gene. This resulted in offspring with healthier weights and normal coat colors, demonstrating how maternal nutrition can directly impact gene expression and disease susceptibility. In humans, the case of the Pima Indians of Arizona offers a poignant illustration of how lifestyle changes can lead to epigenetic modifications with serious health consequences. Historically, the Pima Indians had a diet rich in natural foods and were physically active. However, a shift to a westernized diet high in processed foods and sedentary lifestyles led to an alarming increase in obesity and type 2 diabetes. Research indicates that these lifestyle changes have induced epigenetic alterations that contribute to the high prevalence of these metabolic disorders within the population. Epigenetic changes are also evident in the context of mental health. For instance, studies on individuals who experience childhood trauma have shown lasting epigenetic modifications in genes related to stress response and mental health. These changes can increase the risk of developing conditions such as depression, anxiety, and post-traumatic stress disorder, PTSD. Understanding these epigenetic mechanisms opens new avenues for therapeutic interventions aimed at reversing these detrimental changes and improving mental health outcomes. These case studies underscore the profound impact that environmental factors, diet, and lifestyle can have on our epigenome and consequently our health. They highlight the importance of early intervention and preventive measures in mitigating the long-term effects of adverse epigenetic changes. In our next scene, we will delve into the future research directions in epigenetics and how they hold promise for even more groundbreaking discoveries. As we draw to the end of our exploration into the fascinating world of epigenetics, let's summarize the key takeaways and consider some actionable steps you can take in your own life. Firstly, epigenetics teaches us that our genes are not our destiny. Environmental factors such as diet, stress, and exposure to toxins can influence how our genes are expressed, impacting our health and well-being. To harness the power of epigenetics for your benefit, consider adopting a balanced diet rich in nutrients. Foods high in antioxidants, vitamins, and minerals can positively influence your epigenome. Managing stress is another crucial aspect. Practices such as mindfulness, meditation, and regular physical exercise can help mitigate the negative effects of stress on your genes. Avoiding harmful environmental exposures, such as smoking and excessive alcohol consumption, can also protect your epigenome from adverse changes. Additionally, staying informed about the latest advancements in epigenetics can empower you to make more informed health decisions. As research progresses, new therapies and preventive measures will emerge, offering even more ways to optimize your health through epigenetic insights. In summary, while we cannot change our genetic code, we can influence how it is expressed through our lifestyle choices. By understanding and applying the principles of epigenetics, you can take proactive steps towards a healthier future. Thank you for joining us on this journey through the intricate world of epigenetics. We hope this video has inspired you to think differently about your genes and the environment.